Hi, I am Andrew Ribeiro, and I am happy to talk to you about our study on how promoters in E. coli regulate and evolve the level of extrinsic noise to which RNA and proteins are subject to. This study, conducted at Tampere University of Technology by the Laboratory of Biosystem Dynamics, was recently published in Scientific Reports with the title Rate-Limiting Steps in Transcription Dictate Sensitivity to Variability in Cellular Components. It is well documented that genetically identical cells exhibit different gene expression rates due to, among other reasons, having different numbers of cell components, such as RNA polymerases. Also, these components are inherited in division via stochastic partitioning, which causes a significant lineage-to-lineage variability in RNA and protein numbers. Interestingly, for unknown reasons, this extrinsic noise, as it is called, does not affect all genes uniformly. That is, some genes exhibit weak extrinsic noise while other genes exhibit high extrinsic noise, although the sources for that noise are the same. In this work, our goal was to explain why does this noise differs between genes and what is the mechanism responsible for these differences. To conduct this study, we made use of state-of-the-art single-cell measurement techniques such as MS2-GFP tagging of RNA molecules, which was generously provided by Ido Golding, along with image and signal processing methods that we developed for this purpose. If you're interested in this, please check our website where they are freely available. In general, we proceed as follows. We image cells, we extract information on when new RNA molecules appear in each cell, and from this, we can calculate RNA numbers and thereby measure the lineage-to-lineage diversity in RNA numbers over time by calculating the squared coefficient of variation of single-cell, single-lineage RNA numbers, which is a measure of noise in RNA production. First, we start by presenting empirical evidence for the existence of a mechanism regulating, somehow, the variability in RNA numbers due to extrinsic noise, that is, noise that is due to cell-to-cell diversity in cellular components. For this, we compared the RNA numbers in different cell lineages when having the same promoter but different inducers, and when having the same inducer when applied to different promoters. Visibly, from the plot there, you can see that both factors cause the variability in RNA numbers between lineages to differ. Given the observed diversity between RNA numbers in different lineages and the different degrees of such diversity between promoters and induction schemes, we formulated a hypothesis. You can see the chemical reaction of transcription. It has a multi-step process where it starts with a closed complex formation and it follows the open complex formation, after which a fast process of elongation takes place to produce the RNA. Now, if you look at these reactions, you will see that the speed of the first step, the closed complex, depends on the RNA polymerase numbers. Meanwhile, the speed of the second step, the open complex, does not depend on the RNA polymerase numbers present in the cell. Thereby, we took the example of having a gene where the first step is very fast and the second step is very slow. And let's imagine that the total duration of production is 1,000 seconds. And the first step is 100 seconds and the second step is 900 seconds. If I double the speed of the first step, so going from 100 seconds to 50 seconds, the total change in the gene expression in the RNA production rate will be going from 1,000 to 950 seconds. So that's not a big change. Meanwhile, if you consider the opposite, that is, if the first step takes 900 seconds, for example, and the second step takes 100 seconds, and if I speed up the first step to double, because I change the RNA polymerases to double concentration, then what we will get is going from 1,000 steps in duration, 1,000 seconds in duration, to 450 plus 100, that is 550 seconds in duration. That is, in the second gene, the same change in RNA polymerase concentrations led to a big change in the transcription rate, while in the first gene, it only led to a very small change in the transcription rate. That is, the change in the overall transcription rate due to changes in RNA polymerase numbers depends on the relative duration of the first and the second step. The same phenomenon occurs, in our opinion and based on our hypothesis, in the cell-to-cell variability in RNA numbers production, because if we add variability in RNA polymerase numbers, this variability will affect the variability of the kinetics of the first step alone, and thereby the same phenomena of causing a big effect in cell-to-cell diversity in RNA numbers only and mostly for genes whose closed complex is relatively much slower than the open complex. Let's now consider that instead of changing RNA polymerases, we're changing the concentration of inducers. Inducers also have cell-to-cell variability in concentration, so we will have similar effects. The more complicated fact about inducers is that the inducer may affect the closed complex, the open complex, or both. And the effect on one may be stronger than the effect on the other step. So we need to determine the steps that the inducer affects. But let's now consider the assumption that we do know which steps are being affected. If this inducer affects mostly the closed complex, then a similar phenomenon to the changes in RNA polymerases will occur. Meanwhile, if the inducer affects mostly the open complex, then it's the opposite. That is, it will change, in fact, the CV square of the RNA numbers between lineages, but it will do so by changing the open complex, not by changing the closed complex. In this paper, we have produced models for the inducers as well, and of what occurs as we change the inducer in the CV square of the RNAs. And then what we did was we exactly changed the inducer concentration, and then we confront each one of the models with the data to determine which one is the most appropriate model. We then set out to prove our hypothesis. For this, we conducted measurements of the duration of the open complex formation in the 4 cases shown in the table. LECATA-1 promoter subject to 1 millimolar of IPTG, LECATA-1 promoter subject to only 0.1 millimolar of IPTG, then subject only to 0.01 millimolar of IPTG, and when subject to 1% of arabinose. To do these measurements, we first set conditions where the RNA polymerase concentration inside the cells differs significantly between such conditions. You can see on the left bottom plot that we were able to, by putting the cells in different media, to relatively to the first media, change it almost by 60%. Given that the cells are in such different conditions, we measured in each case the total rate of RNA production. And from that total rate of RNA production, we got 3 points, which we plot in an example tau plot, where what we're plotting is the inverse of the transcription rate as a function of the inverse of the RNA polymerase relative concentration. And what you can see is that by placing a line of best fit to the measurement results, one can estimate what would be the rate of RNA production if the RNA polymerase concentration was infinite. That is the point where the line crosses the y-axis, because this corresponds to an infinitely fast closed complex formation. Thus, the remaining time between RNA production rates in the y-axis must be due only to the open complex formation. As such, we obtained for each promoter and each induction scheme the relative durations of the open and the closed complex formation that is relative to the total time interval taken between transcription events. In this slide, we present the empirical validation of our hypothesis. To do that, we considered a model where the inducer either affects the closed complex, the open complex, or both. To test the effects of it, we placed the cells containing the LACADA1 promoter under different induction conditions at 0.01 millimolar of IPTG, 0.1 percentage of arabinose, 0.1 millimolar of IPTG, and 1 millimolar of IPTG. And then we asked the model for those concentrations which correspond, according to the measurements, to a given value of TCC over delta T, what would be the noise in RNA production. What you see there are the three possible models. So the black line is the model where it is assumed that the IPTG or arabinose levels affect the closed complex formation. As you can see, that model, the black line, does not fit at all the 4 experimental data points. Then we considered the blue model, where the inducers affect the open complex formation. That model fits pretty well the data for the lower concentrations, but it does not fit the data very well for the higher concentration of IPTG. Finally, we considered a model where both steps are affected by the inducer. That model, the green line, fits very well the experimental data. So from this, we conclude that the inducers, in the case of LACADA1, affect both the first and the second rate limiting step of the transcription model. But notice that the first step, it appears that it's only affected for the higher levels of concentration. That is, for lower levels, it appears that the open complex model suffices to explain the empirical data. From our results, we can make two main conclusions. The first conclusion, as said in the previous slide, is that the model that best fits the data is the one where the inducer affects both the closed and the open complex formations, but mostly the open complex formations. That is, the effects being important on both steps only occurs at the high concentrations of the inducer. At the low concentrations, we can fit the data well with the simpler model where the inducer only affects the open complex. This is important because in cases where the simpler model fits the data, we should select the simpler model. This is because, of course, the more complex model can tune both variables, and thereby it will always fit the data at least as well as the simpler model. Finally, our main conclusion is that in general, the effects of cell-to-cell variability in inducer concentrations, or for example in RNA polymerase concentrations, on the variability between lineages in RNA numbers can be tuned by the ratio between the closed complex formation and the total duration between transcription events, or of course the ratio between the open complex formation and the total duration of transcription events. Because of that, this variability between lineages is both subject to regulation, because the speed of these steps, the rates of these steps, can be altered by inducers, and it is evolvable because the rates of these steps are promoter sequence dependent. Finally, we would like to note that in this presentation, we only presented some of the key conclusions from our work. There are other interesting results in our publication that we hope that you are interested in and read. Some of these results are shortly described in this last slide. If you wish to obtain information on how we reached these additional conclusions, or if you have any questions, please download the manuscript and supplementary material on our group's website or contact us directly. Thank you very much for your attention.